Imagine a world without antibiotics. It isn't hard to do. Take a look back into our recent past where an infected limb could result in an amputation, and people could die post-operatively from what is now considered to be minor surgery. Before antibiotics, it was not unheard of for healthy people to die of diseases such as pneumonia, and the recovery period for many illnesses was much longer. These days, commonplace infections are quickly and easily treated with a simple visit to your GP, but now, after half a century with antibiotics, we are looking at the prospect of again having to live without them. Antibiotics work in a number of different ways. Beta-lactam bacteria such as penicillins work by inhibiting cell wall synthesis, while some inhibit protein synthesis, nucleic acid synthesis, and the synthesis of folate. Some will also disrupt the phospholipid component of the cell membrane in gram-negative bacteria, leading to an increase in the cell permeability and in turn cell death. Antibiotic resistance occurs when bacteria have lost their susceptibility to the effects of a particular antibiotic, which renders the treatment ineffective. Some bacteria are naturally resistant to particular antibiotics. However, bacteria may also become resistant in two ways. Firstly, they can acquire a genetic mutation, or secondly, they can acquire resistance from another bacterium. It is a perfect example of survival of the fittest. Bacteria that can resist a particular antibiotic have a greater chance of survival than those that are susceptible. Susceptible bacteria are killed or inhibited, resulting in selective pressure for the survival of the resistant strains. Some mutations enable bacteria to produce enzymes that inactivate antibiotics, while other mutations alter or eliminate the target molecule that the antibiotic attacks. Some close up the entry ports that allow antibiotics into the cell, while others manufacture pumping mechanisms that export the antibiotic back outside so it never reaches its target. The most common form of resistance comes from bacterial production of beta-lactamase, an enzyme that cleaves the amide bond of the beta-lactam antibiotic ring before it can interact with the penicillin-binding protein on the cytoplasmic membrane of the target bacteria. The beta-lactamase gene can then be transferred from resistant bacterium to non-resistant bacterium by a process known as conjunction. In some countries and online, antibiotics can be purchased without a doctor's prescription. Patients often take antibiotics to treat viral illnesses like the common cold or do not follow directions given by their doctor. Global travel and medical tourism are also issues, as well as lack of new antibiotics available. Antibiotic resistance not only makes bacteria harder to kill, it can actually make them stronger. Once a bacterial strain is resistant to several different antibiotics, it is referred to as a multi-drug resistant microbe. When there are virtually no antibiotics available left to fight a bacterium, it is said to be pan-resistant. These strains are becoming more and more common not only in hospitals but the community at large. Methicillin-resistant Staphylococcus aureus, or MRSA for short, is commonly found in hospitals and is resistant to many antibiotics. In the public, MRSA infections are usually in the form of skin infections. In medical facilities like labs and hospitals, MRSA can cause deadly bloodstream infections and pneumonia. These gram-negative bacteria can survive for a long time on surfaces in the hospital and can enter the blood through catheters, open wounds, and ventilators. Drugs used to treat gram-negative bacteria come with serious adverse side effects such as renal impairment and elevated creatine levels, making them difficult to treat. Another major health problem is the multi-drug resistant tuberculosis. TB is spread from person to person through air. It affects the lungs, however, it can affect organs such as the brain or kidneys. This disease is caused by bacteria that have become resistant to rifampin, which was previously used to treat TB. So how can we slow down the escalating emergence of superbugs? Good hygiene is very important, as this prevents the spread of infections so that fewer antibiotics are required. It is vital that we educate people on the importance of compliance, as many people do not finish their courses or take drugs as prescribed. It is also up to medical professionals to ensure that antibiotics are reserved for cases that really need them. These are simple steps that can be followed by everyone. However, hand washing and related measures can only control the spread of resistant bacteria. Vaccines are being developed to immunize people against antibiotic resistant bacteria. These, along with pre-made antibodies, can be given to people who are at risk or infected with drug-resistant microbes. Farms in many countries use antibiotics for the promotion of growth and prevention of disease in livestock. Resistant bacteria work their way along the food chain from animals such as chickens and eventually end up in the blood samples of infected patients. This is known as the farm-to-fork phenomenon. By stopping this overuse of antibiotics, this helps in stopping the growth of resistant bacteria before it reaches the population. Collecting data on the use of antibiotics can determine specific problems and where they arise. An example of this is a study conducted by the European Union over 15 years, which showed that countries with the highest use of antibiotics also showed the highest proportion of resistant bacteria. This study demonstrates the strong association between antibiotic use and resistance and also identifies where corrective intervention is needed. As well as researching and developing new antibiotics to combat resistant strains, developing antibiotics that interfere with the actual mechanisms that give the bacteria resistance, rather than killing it, is an alternative way of dealing with the problem. An example of this interference is stopping the duplication or movement of the bacteria's genetic material, which eliminates the problems of these resistant genes and mechanism of transferring to each other. Another approach is the development of drugs that interfere with the mechanisms that give bacteria its resistance. This can be done by stopping the duplication or movement of genetic material from one bacterium to another. Beta-lactamase inhibitors such as clavulanic acid can also be given in combination with penicillins to prevent inactivation of the antibiotic.